The ER membrane of yeast or higher eukaryotes has a protein complex known as SEC63 complex. The SEC63 complex is embedded in the ER membrane. The SEC63 complex is tetrameric in structure. In the vicinity of SEC63 complex, a translocon is present. This translocon is also embedded in the ER membrane. The cytosolic unfolded polypeptide chain having a N-terminal sequence translocates through the translocon. As a result, the N-terminal segment of the polypeptide chain enters the ER lumen. In the ER lumen, signal peptidase then cleaves the signal sequence just as the co-translational translocation. As a result, a cleaved signal sequence is formed. This NH3+ marks the N-terminal domain of the cleaved polypeptide. The cleaved polypeptide chain further slides inwardly. In the ER lumen, a protein called BIP is present. BIP is a member of HSC70 family of molecular chaperones. Like other members of HSC70 family, the BIP has a peptide binding domain and an ATPase domain. The luminal ATP then binds to the ATPase domain in the BIP. The ATP-bound BIP then interacts with the luminal portion of the SEC63 complex. Due to this interaction, the bound ATP undergoes hydrolysis to produce ADP and PI. The PI is then released into the ER lumen. Due to this ATP hydrolysis, the BIP undergoes a conformational change. This conformational change promotes the binding of ADP-bound BIP to the exposed polypeptide chain in the ER lumen. As the SEC63 complex is located near the translocon, BIP is thus activated at sites where nascent polypeptides can enter the ER. Binding of ADP-bound BIP to the luminal portion of the polypeptide chain prevents the backsliding of polypeptide chain out of the ER. When the polypeptide chain further slides inwardly, more of the polypeptide chain is exposed on the luminal side of the ER membrane. To this exposed part of polypeptide chain, another ADP-bound BIP binds with the release of a PI. Like this, many ADP-bound BIP bind to the further exposed part of the polypeptide chain. For simplicity, only two ADP-bound BIP molecules are shown here. As a result of binding of many ADP-bound BIP molecules to the exposed part of polypeptide, the entire polypeptide is drawn into the ER lumen within a few seconds. The BIP molecule then spontaneously exchanges its bound ADP for ATP. Due to this exchange, the BIP undergoes a conformational change. Due to this conformational change, the ATP-bound BIP is released from the polypeptide chain. Like this, each BIP molecule spontaneously exchanges its bound ADP for ATP. Due to this exchange, each BIP undergoes a conformational change. Due to this conformational change, each ATP-bound BIP is released from the polypeptide chain. These freed ATP-bound BIP molecules are now ready for another interaction with SEC63 complex. The polypeptide chain released from the BIP molecules then folds into its native conformation. Now, important point, the SEC63 complex are required for co-translational translocation. Please like, subscribe and share.